live streaming have started. I think we can start.
Yes, sir. You're live now. Please start.
All right. Very good morning, everyone. Welcome to all the students who are joining today for MOOC live session. All of you have started the week 1. I hope you started very well your academic semester, and you are learning some new things in the areas of proteins and proteomics, and going forward more about proteogenomics as well. So, idea for live session is to interact with you more, but at the same time give you some flavor of what we do in our research labs here, and give you some idea for something more thematic. And therefore, today we are going to talk to you about gel-based proteomics as a focus. That will be common to all the courses. So today we are going to talk to you about how proteomics can be done using different type of gel-based approaches. And we'll show you some of the live experiments and demos from the lab so that you're more familiar with. The idea is like, the field of proteomics started almost 50 years back when first time scientists Close and Farrell, they were the one who tried to separate the complex protein mixture based on 2 properties, looking at their molecular weight and isoelectric point. And that became 2-dimensional electrophoresis. So that was 1975 paper. And almost 25 years from that time till 2000, the field enjoyed very much of doing proteomics using 2D gels. But then more and more newer approaches started coming in. Some of the label-based approaches came, like ICAT, using mass spec-based approach. Similarly, then DICE came, which is fluorescence difference in electrophoresis method for 2D gel. Those approaches told that there could be much more reproducible, reliable, and more high throughput method of doing quantitative proteomics or differential proteomics. And slowly then gel-based proteomics started reducing the impact, especially 2D gels. And going forward, DICE became more important. And eventually, after 2005 or so, shotgun proteomics using the LC-MS became more prominent. And currently, the quantitative proteomics is heavily done using different type of mass spectrometers, especially the timsTOF, the Orbitrap. Some of these are the most robust instrument and method, which we'll talk more in the next live session. But idea I want to give you bird's eye view how the field really evolved over the time. At the same time, if you think about whether gel-based proteomics totally stopped, no. Gel-based proteomics is the most essential component for all the proteomics work, whether we do quantitative with gels or not, but still many things are being done on the gels. For example, when you're doing protein extraction, very first thing which you do, you want to do quality check of your protein samples, and that you run on the SDS gel to ensure that you see very good uniform separation of the protein, and those proteins are seeing in the on the gel properly. So those kind of things are being done on the gel very, very routinely. Also, if you're purifying the protein, you always want to cross-check the molecular weight on the gel. Many times people are looking at some sort of impurity detection, some sort of contaminant, looking at that also on the gel. Many times, when you're purifying the protein, you are expecting some band, but something else comes as well. And those could be co-purified protein, which you're interested in, are they contaminant or something very interesting interactors. So one would look again on the gel. Sometimes the complex samples, like, for example, urine sample, cerebrospinal fluid sample, or some other salt-grown sample, which are very difficult to run directly in the mass spec, one could actually run them on the gel and excise the gel pieces. And then from those gel pieces, you can do the in-gel digestion and put it together to make it proteome. So again, you can see the utility of gel to do the complex proteome analysis. It all depends on what your goals are, what your objectives are for doing a given proteome experiment. But regardless of that, you're always going to use the gels in your lab as the very first benchmark for looking at your sample quality, and whether non-specific things are not there on the gel, those things you want to ascertain before you move on to the actual proteomics workflow of doing the digestion of the protein or looking at LC-MS based analysis. this is just, one way of thinking about it, how one could look at the proteome analysis. But many times you are looking at, say, interactome analysis. You're looking at pull down assay. You're looking at, purified complexes, and you want to identify every single component of that complex, which is not a whole lot of protein, but those could be 15, 20, 30 protein. And you will still like to separate them on the 2D gel so that you are able to distinguish those very well based on the molecular weight and isoelectric point kind of properties, and then use those information to then find out which are the potential interactors of your given complex or given protein of interest. So again, you can see utility of gels can be very high. So for many applications, you will still see different type of gels being used. They're very commonly available in all the labs. And again, if your goal is to do good proteomics, your very first thing should be to look into the protein extraction protocols and protein separation and quality check, which is done on the gels. That is your first benchmark, gold standard. Once you have done that, then you will quantify the protein. You will now look at those quantification and you proceed for the protein digestion to make the protein into peptide form. You clean up those peptides, and now you can do different type of analysis using the label-based or label-free approaches. And then you can do different type of mass spec-based investigation to find out what could be differences across those protein samples. So this is just kind of, bird's eye view, but I hope I have made a very balanced perspective of while the gels and 2D gel cannot be used right now as the first gold standard for doing quantity proteomics, while LCMS can give you much more robust and reliable and reproducible data at very high throughput level in a very short time. Nevertheless, the gels still remains the very first step of the quality checks, and they are the best thing for your biochemistry to be understandable and to take forward for your proteomic analysis. So today's session, we have Dr. Arya Banerjee. He is a PhD from IIT Bombay. Also, he was awarded as a Best Thesis Award from IIT Bombay. He is a TA for this course, and he's very experienced both in the classroom teaching here at IIT Bombay and the MOOC courses. So you are very privileged to have him in this course. He is coordinating your Telegram group and the forum activities. So today, Arya, along with our other TAs and the students from the lab, are going to take different concepts of the gels and show you the live from the sample prep to different type of analysis directly from our lab. So Arya, over to you, and then we'll continue with some of the hands-on sessions.
Thank you, Prof. So, just a minute, I will share the.
So we will spend less time arguing on the PPT because now timing is short. So just give a quick overview, but then we move on to sample prep. All right. So while Arya is getting ready, the gels has to still go to mass specs. Even when we say gel-based proteomics, alone they are not sufficient to give you the proteomic data. So we will still take them forward on the mass spec, regardless whether you have the bands from the gel, which you want to identify what those proteins are. So you will still look at the in-gel digestion of those proteins. And likewise, if you are looking at any type of complexes, you'll again take those spots and do in-gel digestion and look at those interactors. Arya, over to you now. Unmute yourself.
So welcome to the gel-based proteomics in mass spectrometry. So what question we are addressing is why seeing protein still matters. Like mass spec can help you to quantify proteins, but the gels can help you to understand how we can visualize the protein well. So this is one of the information which have been shared by the Professor Sanjeeva. From 1975, the 2D gel electrophoresis came into existence to the mid 2000, where the level-based SILAC, TMT, ITRAC, and the level-free methods were developed, to the current era where we are monitoring into the single-cell proteomics, and to the more advanced version of the proteomics where clinical proteomics, translation research, and biomarker research is being conducted. Okay, so if the modern MS is so powerful, then why do the still researchers need the gels, like the use of the gels? So in the left hand side, you can see the fragmentation, which will help you to answer more. And the right hand side, we have the gel based proteomics. You have a traditional 2D gel image spectrum where you have the molecules are separated first on the basis of their isoelectric point, followed by their molecular weight. So these are the same proteins. So basically these are same proteins but different forms. Mass spec can help you to identify which proteins are present, what are the differential regulation of the proteins, their fold change, their significance, and how many peptides are each identified from the each of the M by V values which have been tabulated. But what gel-based electrophoresis answers are most, are there any isomers? Okay, you can clearly visualize that. Are there any PTMs which you can quantify, but you can easily clearly visualize in the case of the mass spec? Are there degraded products? Are there contaminants? And is there a heterogeneity caused? So this is the mass spec. So mass spec identifies the proteins, and whereas the gel-based electrophoresis visualizes the proteins. Why clinical samples are difficult? Urine, as already Professor said, I will just give a brief. Urine consists of high salt content. Saliva consists of high mucin content, plasma, wide range, like very masking proteins, like the dominant proteins are present. You have to remove or deplete it to make the significant protein to be expressed, lipids, metabolites, and various detergent interfaces. When we go to the purified molecule, when we go to the section of the pure molecule characterizing, it needs the confirmation of the purity and identity, whether the protein is truncated or degraded, whether it is aggregated, whether the isoform and the charge are present. Finally, in case of the protein and interactors identification, like how the interaction is lost during the denaturing conditions or the native condition, these all things you can visualize along with the host cell proteins and the keratins. So yeah, impurities and contaminants can also be identified from the same. So coming to the, so I will just give you, this is how a 2D gel based electrophoresis flow. Okay. So 2D gel electrophoresis flow. In two-dimensional electrophoresis, you undergo isoelectric point means separation, followed by separation on the second dimension on the basis of the SDS stage, like molecular weight. And finally, you visualize the pot with the decider, like decider. So what are the advantages? It already removes the salt. Separation is according to the PI. Reduction of the sample complexity is minimal, and also visualization of the intact protein is happening. A major advantage of one of the 2D gel electrophoresis is the dyes, where we are using the Psi dyes, the minimal labeling Psi dyes like Psi3, Psi5, and Psi2 to create a multiplexity. So you can see you can analyze more than like 2 or 3 samples, you can complex it, and based on their different fluorescence, like you follow the same routine procedure, separating first on the basis of the isoelectric point, followed by the isoelectric focusing, and finally you see that like the spot analysis and the quantification is very important. It has a wide clinical representations, like most of the papers have been shared with you in your study material. It can help in, means most of the studies have been done for the breast cancer, colorectal cancer, Alzheimer's. Parkinson's and Alzheimer's are the broadly used one for using the dye like 2D. In case of the biopharmaceutical industry, biopharma comparison, vaccine development, recombinant proteins characterization is very much stable. In case of agriculture, you look for drought stress resistance, pathogen resistance, and crop improvement, followed by the microbiology antibiotic resistance, which is very common, and microbial adaptation. So I will just give you what happens, the first part, which is the protein sample preparation, like how you prepare the samples for running the samples in the dye. So you can take the desired cell pellet, tissue, or the serum samples. First, you have to denature it. Okay, you have to lyse it in the denaturing condition. For that, you need to lyse this tissue. So urea lysis can be used, one of the most global, like gold standard lysis buffer. Along with it, you add a protease cocktail inhibitor, followed by the quantification with the Bradford, which helps you to identify how much amount of protein is present. Like this is the sonicator. We will be showing one of the cutting edge technology, which is the bit beater. You go for the Bradford estimation. And once the Bradford estimation is done, in the routine lab, what we do a general SDS page to check the quality of the proteins. Okay. So this is how a Bradford concentration standard curves looks like. R square of 0.99 is good enough. And with the straight line equation, you determine the curve. Rather, after going for this, like the Maldi, like after going for the 2D diage, what bands I have shown you, like if these are my bands of interest, you can excise these spots or these bands and you can do an in-gel digestion, which can be followed by the Maldi analysis. Like this is the protocol for that. So now we will move on to the demonstration of the sample. So
All right, so after some overview, now I think the main agenda is to give you the actual demonstration in the lab. And Arga and the TA and the team of Proteomics lab is going to give you more like cooking show, some of the key steps involved in from sample processing towards moving to the gel. Arga, all right, you're not audible though. Arga, you're saying something?
yeah, is it audible?
Now it is audible.
So yeah, so what apparatus you can see here. So this is the proper clinical bench setup where we follow the aseptic conditions with the lab coat and all those things. And only one criteria keep in your mind is like you have to be cold and precise and very clean. Like many contamination can happen, either keratin contamination, wear gloves while handling the samples. So I will start. So Sairaj is with me. Sairaj, you can take the samples. So this is how the sample look like. So this is a magnetic state bearer, which is generally called as the bead box. It lies the sample in, it lices the sample under the magnetic array, like a magnetic field. You place your beads, you place the pen drop tubes in this sample. You put the samples, and here is the matrix where you can set. You can select for the low. You can select it. You can select it for the low, high, as well as the standard, like whichever mode you want to choose. Okay. So now is currently it is set in the mode, and once this is done, you can lock it, like you can unlock it from here. And once the locking procedure has been started, you can start the samples. Okay, so it will be doing the lysis in 10 minutes. So I will just stop it for the demo purpose. Once the sample is being done, you quantify your samples with the help of Bradford. So this is a standard plate. I have put the colors like a 96 well ELISA plate you can use. You can place the samples your known or the standards in one of the well, followed by your lysate, and then you put around 200 microliter of the bat, and then you put around 200 microliter of the Bradford. So this is how a plate inlet looks like. So this is done, so once you have gone for the recommended protocol of the 10 minutes incubation, so this is a microarray plate reader. What you are seeing, you you can put your plate, inside the scanner. So this is how the scanner set is putting, and you can do a reading. So this is how the matrix looks like. You can set the plate layout. Okay, and you can do the reading. Okay, so once the quantification is being done, you can plot a straight line equation, what I have shown. You can check the standards, you can check the calibrants, and once the reading is being known, the unknown concentration, you can start for loading of the gel. So this is how a standard SGS gel apparatus look like. In two-dimensional gel electrophoresis, this becomes the second dimension parameter of the analytical separation. And this is how the gel can cast it. Depending upon what is your objective of the study, whether you are doing a pharma application or any other application, you can set your SDS page from 8% to 12%. And also, like, you can change into a gradient base. If you don't want to cast the gels, ready-made pre-casted gels are also available. So as per your need, you can cater the needs. So this is how, like, once this is being done, so this is a gel casting, this is the running apparatus. So what is the properties of the SDS page? The overall negative charge is there, where molecules get separated on the basis of the molecular weight. The closer to the lane, and the larger to the lane of the larger charges, you can find and correlate it accordingly. So once the gel is being run in the apparatus, uh, so here you can find, so this is a clear demonstration of the gel. There is a marker, there is a marker. So there is a marker you can find, like this is the, so there is a marker what you can find. Like, these are the standard known markers of the known molecular weight. I am exposing it without gloves, but always wear gloves because this is the one of the run gel. So you can find, like, these are the molecular weight which have been aligned. Like, you can decode the molecular weight with standard markers. These are my known samples, and this is how the prominent the gel band patterns look like. You can put it, you can scan it in the scanner, you can scan it in the scanner, which is a typhoon scanner. And back to you, Prof. By then, I can get to the this specific.
All right. So again, the sample preparation becomes very important aspect for any type of proteomic analysis. Timing was not sufficient for us to show you the complete detail. However, at least you got some idea that there are newer methods coming up which can help you lyse your samples better, especially bead beater. You can always try more than one method so that your lysis is very efficient. Because if the lysis is proper of your cell, then only you will be able to get everything out from the cell, and you can now see how best you can separate the proteins along with other contaminants. Contaminants, I mean here now the carbohydrates, lipids, nucleic acid, they're all contaminants for proteomics. So we have to remove them all out. And eventually the most culprit is the salt that has to be cleaned. So after we have done that, then only we have a decent sample to proceed forward. And a good benchmark is the gels, which is the LGS page gel, which you just see the bands, which tells a lot of information that how good my extract is and how well we are able to separate those proteins and are there any impurities, etc. This could be your first beginning. Sample prep should be ideally your biochemistry and your, depending on your objective of your experiment, you should decide the sample criteria. SGS page or the gel testing of your qualitative analysis should always be there, irrespective of whichever protocol you're following. But the next thing which we're going to talk to you is about 2D gel, which is the next in line, about showing in which way you can separate the complex protein mixture based on isoelectric focusing. So now again, the team is going to briefly show you the steps involved in doing 2D. I'll also join in the lab shop.
So thank you, Prof. So what you are seeing is an ETAN IG43, which is, as in the figures I have already shown, like the first dimensional, you go for the isoelectric focusing. We have our demonstrator Nitin with us. So Nitin, you can start the steps.
First, we can prepare the protein using a rehydration buffer. We can load the protein sample with rehydration buffer in a rehydration tray. This step going for 10 to 16 hours for complete rehydration and complete uniform protein distribution. So then we transfer our strip in our Eton IPG4 tray for the isoelectric focusing. We fix the paper wick and electrode on the strip, and this step is going for this step is going for 5 to 8 hours.
All right. So, say idea is, here a lot of optimizations can be done, depending on your goal. If your goal was to look at only simplified complex, which I said to you, like for interactomics, so that we should just look at the smallest strip, like what you see here is a 7 centimeter strip, which is very small. But if your goal was to look at a complex proteome, which is having 5,000 protein, you should look at the very long IPG strip, which is 24 centimeter long. And that's where it will take you several hours. If you can come zoom in here. And so now this can be the full length of the tray. And the best part of this particular setup is that it's very flexible. You need not to, you can change the electrode and need not to use, different trays for that matter. You can use the same tray and use different length. So as you've already seen that there is a 2 paper bits which is already put on the sides. They are absorbing all the salt. So during the isoelectric focusing run which is happening, any contaminants are usually coming out from the strip and those are being absorbed towards the edge on the these paper bits. And that's where initially we keep very low voltage in the setting. We keep 200 to 500 volt for a few hours so that any kind of salt and things get absorbed in the edges. And over the time, then we ramp up and we change the voltage. We initially go maybe 2000 volt, and then after a few hours, then we change to 7 or 8,000 volt, and we try to achieve the volt hour, not only voltage. We look at for the small strip, we want to achieve 75,000 volt hour, for example. For the large strip, we want to achieve maybe 125,000 volt hour, and we give the time that many hour the instrument can be kept for that voltage to achieve the volt hour. So therefore, depending on sample movement, sometimes this IF can finish in 20 hours, sometimes it can get 24 hours. And but you want to achieve a specific volt hour depending on your objective and the type of IPG strip. But this is the, I think, this field that as you can see still in our lab, this is very valuable resource. These kind of things are always have been the gold standard for doing proteomics. Over the time, people realize that running the gel is easy, but doing the reproducible gel of maybe 200 samples is not that easy. And that's where there was a movement and shift from the 2D gels moving to the shotgun proteomics approach. But this instrument, the first approach, is looking at the isoelectric focusing separation. I'd like to again remind you, similar kind of instrument setup can also be used even if your goal is not to do the gel-based proteomics. For example, you can use the liquid IEF, liquid isoelectric focusing, same instrument, but you are going to use the cups. Those will be plastic cups where your entire protein will move across different cups with the IPG strip. And you can now take those fractions of your protein from different pH value. For example, between 4 to 7 pH, you have 20 cups. Now the proteins have moved to the respective cup, and you are going to take those fractions which are specific to those type of PI value. And then now you can run those in the mass spec, and then you pool the data. That is one way of fractionation. You can fractionate your complex proteome using one principle, which is here with the isoelectric focusing, or the PI value, isoelectric point of the protein. While there are other principles which we use in the chromatography, which is reverse phase, another affinity, and many other ways you can do with the chromatography also. But many times people are still, if the goal was to look at the complex proteome separation and look at all the protein of a given sample, you should fractionate very well. And 2D gel, the first part of 2D gel, which is the isoelectric focusing, that setup is still being used as the liquid IEF to do the protein separation. This is the first step. After this step, now we have to prepare our IPG. So let's say proteins have now fixed and they have moved to the PI value, which is exactly or the pH value, and there that is the net charge is 0. Now you will take this particular IPG strip and you are going to place in the tray like this, right? And so that IPG strip you are going to place it here, and you are going to clean up that IPG strip with the different equilibration solution. For example, think about your SGS page. When you are going to run the SGS page, your proteins are going to be boiled, right? You're going to be preparing your complex sample so that there is no disulfide bond, so that reduction alkylation you do before you run on the SGS pages, the proteins get separated well. But here in the 2D gel, we don't have that opportunity. So after coming from IEF and before going to the second dimension separation of molecular weight, we always go for in-between step, which is known as equilibration step. And that step happens in the tray like this here, where you can place your one strip, IPG strip, in the equilibration solution 1, which contains the DTT along with the glycerol and the tris, etc. And then the solution 2, which will be the iodostamide. So those things we can also maybe show on the screen here, like this step, which is we are going to clean our IPG strip in different equilibration solution 1 and 2 to make sure that there is no disulfide bonds are there with DTT, and there is no reoxidation happening, no alkylation happening with the iodostamide. So that we just got a feel of it. Again, we can talk back here. So these steps are very critical, and you need to ensure that around 15 minutes of time is being used for showing both of these steps of 1 and 2, right? So once we have done that, then we come to the take our IPG strip, and we have to clean those IPG strips into different, on the paper wicks and the kind of tissue paper, and then we move on to the second dimension separation. What you see here is a very small 7 centimeter gel sector, but ideally, the goal was to look at the large 2D gel separation, we can separate those on the P10 dart or one of the Ruby setup, which you can bring in here and we can show. So then we can do those separation on the large units as well. Again, it depends on what our IPG strip length is. If the length is 7 centimeter, this gel is good enough. If the IPG strip length, as I mentioned to you, is very large, could be 24 centimeters. So then we are going to utilize for 24 centimeter IPG strip a big unit, which is known as a Cal-Dunt unit. And in that unit, we keep 6 gel separations simultaneously. So, for example, what you see here is the big setup. This we don't use routinely, we only use that when we have the 6 gels to separate. So, if you can come closer, Angela, what we can show here, that there could be 6 large gel which can be kept in this particular setup. So now think about you have 24 centimeter long strips from 3 control samples and 3 treated samples, and you are going to separate them simultaneously in the second dimension using this big unit. So that's where I think this setup becomes very attractive, because you are reducing the gel to gel variability and separating very complex proteome. So on this kind of IPG strip, you can easily go even more than 1 microgram protein, which is sufficient for looking at thousands of protein on the gel. And those you can separate those on this gel. So running these 6 gels together is actually very difficult task, because first of all, casting the gel, I'm sure many of you would have failed already with the 7 centimeter small gel to make SGS page properly. And now if you have to cast 24 centimeter long gel, not 1 but 6 gels, then that becomes a really good practice. And if you can do that, then you're going to place your IPG strip on all the 6. And at the same time, you have to ensure there's no bubble there being trapped on IPG strip. You place your buffer, and now you're going to start the run. But this can give you very decent quality data, which can be actually as comparable as some of the good mass spec based data. So now once you will follow up your run progression, so then on that run progression, you can see where the dive fronts are, and it will take you several hours to run. This can take you good even overnight time, maybe 12 hours, 14 hours, or longer time to separate these complex protein mixture. A 7 centimeter gel can take you maybe just 3 to 4 hours time, but the gel can take you good 10 to 12 hours time. So, but again, the goal was to look at complex proteome mixture separation, but also in this case, we are looking at differential proteomics, 3 control and 3 disease sample at the same time we are running and separating them. At the end, you get sometimes the gels like this, which is the not so great. You have a lot of abundant protein here. Albumin is probably seen on the gel from the serum sample, which means it is masking everything else. But even this gel is giving you a lot of interesting information that there is a depletion requirement, because this part what you see highlighted is actually having the blob of the protein, which means having a lot of albumin and IgG, etc. So you need to clean up these samples and deplete those to ensure that you have the sufficient quality of the proteins from that. So this is the some basics about how one could do 2D gel and even some level of quantitative proteomics using 2D gel. But if the goal was to look at the quantitative proteomics, I would recommend you to look at the labeling approaches. For example, can you take 3 samples and label them with Psi3 and the 3 samples with Psi5? Make the internal standard by combining all of them, and then that can be labeled Psi 2, and then you mix all of them and then do the same separation with the IEF, first step of isoelectric focusing and second step of the doing the second dimension separation. Finally, let's say we have got our complex protein separation on the gel, and you found some interesting spots. So you are going to excise those spots, and you are going to do angel digestion of those so that they are digested and you get a peptide. And now you want to know, you're very curious, okay, what that protein is. I have done my few weeks of work from the protein separation, protein extraction, separation of the gel, doing data analysis, identifying the right spots with the images. They are very significant. Now what that protein is. So then you have to come to mass spec. Now there are different type of mass spec which can be utilized, but right now if the goal is to only identify the protein, very simple, all you want to know what that protein is. So then you can even simply go to multi-top instrument, which can very quickly and rapidly tell you the identity of the protein. So now next demo we are going to have on the multi-top. Let's first show the instrument, then we come back to it. Vertical. Yeah, so now what you see here is the multi-top autoflex instrument, which is one of the type of principle of mass spec. When we talk mass spec, we talk about ionization source. So multi or ESI. Both of them are top generation technology. Both of them got Nobel Prize. This was the very first and robust approach of doing mass spec. So you can do multi-top or you can do multi-top. What we are using currently multi-top. If your goal was to only know the compound mass, multi-top is sufficient. But in this case, your goal is to identify the protein. It means you want to look at the sequence of the protein. Therefore, you need a fragmentation and another mass analyzer. Therefore, you need top. This instrument is multi-top. Now, how this instrument works is essentially we come to you take the angel digested sample, and now you come to this particular plate, multi-plate, which you can see.
So this matrix you can see like A to F and A like 1 to 2. You can mix your samples with the matrix, either synapinic acid or the desired solvent based on their charge. You can spot it and you can insert the plate. Okay. You can put the plate inside the mass spec.
So one thing I'd like to emphasize is that in this particular setup, what you see in the multi, you are actually talking about 384 type of plate, like which is large number of sample you can actually place. So if let's say our goal was to look at you have hundreds of spots and you want to identify them, so very first thing which you'd like to do, I think multi could be a very robust approach, because as you've seen in the plate you can place all those large number of spots and then do that type of analysis on that. Right now what we are showing you here is essentially how to place that plate inside the multi-process. So you can go to the next one. All right, so now I think they'll explain you here that now rest everything, whatever you've seen, the big instrument is all done. You just simply place the your plate which contains the matrix and your peptides.
So now here is the matrix map which is there. You can select like in which matrix your sample is there and you can bombard it with the laser and you will acquire the data. So this is how the data looks like.
Sorry. So this is our peptide, which is exact molecular weight is 3751 Dalton, and we get exact weight 3751 Dalton in our MALDI analysis.
So yeah, so I hope you have seen the MALDI, and in the next set of lectures, we will tell you about the different mass specs also, like how we can develop into this such robust instrument. Just to recap, you have separated the samples from the isoelectric focusing gel, like on the first dimension. Once that is done, you can go for the 2D MS, okay, or you can go for the Newton Dij. Once that is done, if the spots are of important, you can go for the excision, like the angel digestion, which we will explain in the successive classes, followed by if your aim is to insta spot identify the proteins, you can go for the multi-top, and from the multi-top you identify the peaks. Just give me 2 minutes, I will be back. Just give me 1 minute, I will be back with the lectures.
All right, so I'm back here in my office. So I think that the flexibility of giving this live session from the lab, that one could actually go and show you the things right away. I hope you are at least getting a flavor of it, that many theoretical things which we have been talking in the lab, how well one could execute them, and you don't have to always push your boundaries to do every analysis directly on the LC-MS. There could be different applications where your simple protein identification can be done very robust way using the multi-top type of instruments, which actually will take much longer time in your LC-MS-based approaches. So knowing the principles of different type of instruments, knowing the which one is more robust for which application type, is I think is really needed for you to learn as you go along. When you should be looking at in-gel digestion, when you should be looking at in-solution digestion, are again important decisions we made. Now, Arya will talk to you about some of the applications which can be done using this type of approaches, which have been successfully shown that how one could use different type of gel-based approaches and use the gels for different clinical problems and investigate those. All right, Arya, over to you.
Thank you, Prof. So I will just recap, like where were we? You have seen, like I have just given you a brief, but in the successive live lectures, you will find how this you can take out forward for the digestion of the protein samples. Once this is done, we have successfully completed the demonstration of the sample preparation. We have also seen the demonstration of the 2D gel electrophoresis apparatus, how the samples look like. And now after that, in most of the cases, multi-top is being used, whether it is biopharma or some of the applications for the gel excise spots. And we have seen how the instrument means we have seen the instrumentation, we have seen the spotting, we have seen the plate where the multi spot is being there, and how you see the peak. So now let us come to the application, like where 2D Dij has been very much important. So the 2D Dij is very much important. Like you can do, as initially I told, you can do analysis with the urine, you can do analysis with the saliva, you can do analysis with the plasma, as well as the synovial fluid. You can find the papers which I have shared in your study link. So chronic kidney disease identification is being usually done with the help of the, like one of the approaches have been successful approaches was being done with 2D Dij. The 2D Dij identified this disease with the specific protein patterns. It detected the albumin fragments and oxidative modification which was there. So it identified the potential urinary biomarkers from the urine samples. In case of the oral samples, several cytokeratins and the stress response proteins were identified, and the papers is shared with your, like I will share the references with you in a short time, like in the Telegram group. And also, similarly, it is being used for identification of the sepsis, COVID, and autoimmune disease. Okay, now coming to the conclusion, the gel-based proteomics and the mass spectrometric proteomics, they both are complementary. Like both have been related and each has its pros and cons, like one of the niche. When you go for the gel-based proteomics, the intact proteins, like the intact proteins you can find, you can go for the visualization where it is very much important with the help of the two-dimensional gel electrophoresis. But whereas mass spectrometry, you can go for the peptides, you can go for the peptide identification. You can also determine the protein purity with the help of the gel-based proteomics, the proteome coverage, their isoforms, like what kind of isoforms is being used, their quantification, their PTMs and protein complexes. Whereas the mass spectrometry, you can go for sequence information and high throughput sequence. Okay, so this is one of the advantages. In the meantime, I can show you few of the high resolution mass specs where we will be talking and in briefing those things in the next live session.
Okay, all right. So you can come to facility and at least give the glimpse of all the mass spec. Okay, in the meantime, let me share my screen and just show you guys something more. So what you see here is a software known as Matrix Science. Okay. If you have some simple data set which you want to quickly analyze, you can go here and you can, this is actually very freely accessible software which you can utilize. There are multiple options if you want to look at peptide mass fingerprint data, which you can do with the multi-top, which you have just now seen, or sequence analysis or MS ion search. You can look at different type of detail for currently looks like there is some issue with this page. You can look at different parameters and you can look at how to identify the protein. We will have more dedicated session of analysis as we go along. But right now, I just wanted to give you and preempt you that the data which you just acquired can be analyzed with different type of software. And one of this is shown on the screen here. But right now, I think Arya want to show you the instruments in the facility. So we are going back to the lab and showing you different type of mass specs. I'm stopping the sharing. And over to you, Arya. Your voice is not very clear. Arya? Now we can hear you something.
So here you can find like this are the high resolution instruments which have added advantage, complementary advantage to the mass spec, like the gel-based proteomics, what you are having. The evolution of the nano LC, like the nano LC 200, which can go to 6 microliter per minute of flow rate to 200 microliter minute of flow rate with a longer columns of 15 centimeter gradient and have advanced in this field. This is one of the instrument, cutting edge instrument, which is called as the orbital fusion. As you can see, so as you can see, you can place your samples inside. Once the samples have been placed, once the sample has been placed, the samples can be injected, and from the solvent mix, the samples are being tallied and eluted out to a trap column. From the trap column, it goes to the main separation column or the RPLC column, and this is the C18 column which is being used from C18 to the master. And you generally acquire a data like that. Like this is one of the live runs which are happening. So you can easily find what actually happens during the live acquisition of the samples. The peaks, the M by Gs for each MSMS is being detected. We will explain this to a great detail in the next lecture. Apart from this tribrid mass spectrometer orbital fusion, we have Q exactive, which is also an orbiter-based technology in which we perform metabolomics and lipidomic sample analysis. This is an ultimate 3000 series of UHPLC Ultra High, which can work with a binary pump like a quaternary pump and can withstand for 900 bars of pressure like a very robust instrument. You place your samples inside the auto sampler, which is present here. Once you place your samples inside the auto sampler, the injection happens. From the injection, it goes to the column volume, like the column compartment, and from the column compartment, it goes to the like the mass spec, the real-time mass spec, what is happening inside. Okay, and then the sample acquisition happens from the mass spec. Another mass spec which is being used very routinely and frequently in the biopharma sector is the triple quadrupole what we have. So this is a triple quadrupole TSQ audit what we are having. So this is the mass spec instrument like the triple quadrupole, and all this along with that we have a Vanquish HPLC. This is also one of the cutting edge USPLC plus. You can place your samples inside. Like this is the sample compartment where you can place your samples. Once you place the samples in the sample compartment inside, you can run the samples. From the column compartment, it goes to the sampler module. From the sampler module, you finally inject into the mass spec from the TSQ. Apart, yeah, so this is one of the, like these are all of the cutting edge HRMS based techniques what you are having. Back to you, Prof.
Marga, you can show the BLI as well.
Yeah, so this is like one of the added advantage which we will discuss in detail in our upcoming like lectures. This is biolayer interferometry. So what happens is like you can spot your samples or the protein samples into the plate or the matrix which has been present, and based on your detection capacity, like based on their diffraction, like when you have an interactor and when it interactor sits or binds, the refractive index is being changed, which is easily detected, and based on this you can identify the association or dissociation constant when you wash the successive buffers. So this is analogous to the surface plasmon resonance like the SPR, the biolayer interferometry, which is an offset R8 technology, proprietary technology. Apart from that, we have HPLCs and we have NGs. So this is also one of the instruments for understanding or studying the interactors. So studying the interactors.
All right, so thank you, Arjun. So ideally, students, what we have seen that there are n number of technologies which one could utilize to understand the complex proteome. It's not very simple. Therefore, throughout the course, different MOOC courses, we are trying to give you the principles and some of the hands-on and the glimpse of different type of proteomic technologies. From the discovery work, which can be done on different type of mass spec or gels, one could then move on to different type of interaction analysis. I think there are some interesting comments we have on the forum. Let me just take some. So I think Gaurav is saying from the Proteogenomics course about FFP tissue. Can archive samples be effectively used? So it's very nice, curious question, Gaurav. Actually, we like entire field of proteomics and proteogenomics always struggle to see can we utilize those archived formalin fixed paraffin immediate tissue blocks. And what I'm very happy to tell you that with the proteomics, now we are able to optimize the workflows in our lab, which can reproducibly give us more than 7,000 protein in just 45 minutes time. So therefore, it is now possible to do the FFP-based proteomics very well. I'm sure the genomics can be done very well as well. Transcriptomics is difficult to do on the FFP, but overall, I think it's very well achievable. We are also optimizing currently the workflow for metabolomics on FFP, and initial results are looking successful. What is success rate compared to the fresh tumors or the frozen tissue? Again, with the fresh frozen tissue, we are able to get around 8,000 to 500 protein. With the same gradient, same workflow, we are able to get around 7,000 protein when we get the FFP. So it's not that bad, especially if your workflows and everything very consistent. I think now it is possible to do the good proteomics on FFP, and that will be very helpful, as you said, for proteogenomic analysis, because we already can do the genomics very well on that sample. And how to remove the excess salt from the sample? Of course, you have learned some of the very basics of looking at dialysis methods, but similar things we don't use here because we don't have so much protein. So we try different type of filters so that they can bind the peptide, and rest everything we elute them out, wash them off, and then finally we elute out our protein and the peptides essentially. So there are different type of workflows for cleaning up the peptides, and that we utilize with different type of cartridges. And some of those are commercial C18 cartridges and CPEC columns currently being used, which can be effectively utilized for that purpose. I guess I have probably addressed some of the comments which you have here on the forum. Again, if any other students, any other participants who are joining us online, if you have any query, please feel free to ask us. Also, if you have more thought-provoking question, anything else interesting comes to your mind which you'd like to clarify, you can always post in the forum or in the Telegram group. Make it more interactive. Our idea is to not just give you video lecture course, but actually make you feel that we are available to you live from our labs and our office and our classroom to help you give the right training and the good course in the field of proteomics and proteogenomics, so that you can take this learning and go to the next step and do some more experiments and wet lab training, which can make you well prepared to look at some of the good research questions to be addressed. Again, while in this course and in these demonstrations, we are giving you different options of technologies. Our goal is to tell you that if you have a right objective, a right question to address, there could be different technology which you can utilize. You need not to always follow A, B, C, D. You can always go from A to D or C to D likewise, depending on your type of question which you want to address. So this is the first beginning proteomics overview demonstration today we had, which we thought to give you the bird's eye view of different workflows, especially some of the key elements of the gel-based proteomics, followed by protein identification using the multi-Topf-based approach, and that you have seen some demonstrations. Now, if you go back and read those lecture content and tutorials which we are sharing in your forum, you will be much more prepared to understand them better. But we can only teach you as much as you are interested and you are asking questions and you are reading those papers and you are doing those assignments well. We'll be very happy to take your learning forward to the next level. So in the next live session, we will now focus more into the specialized high-end advanced LC-MS-MS-based proteomics-based workflow. And we will show you some of those workflows which can be utilized with the timsTOF and Orbitrap and some of the very cutting-edge LC-MS-based instruments. Till then, please keep reading and keep following our lectures. Any more query? What is the query? Or do you conduct any hands-on or training at IIT Bombay? Of course, we had just conducted Advances in Proteomics Technology APT workshops and conference, which was attended by a couple of hundred students to even 2,500 people came during the entire event. So we do conduct. Probably the next one we are going to conduct a short hands-on specialized training on 15 December. Feel free to join us some of those activities, and we'll be happy to take your learning to the next level. All right. Thank you very much.